Thank you, Cristiano, and good afternoon, everyone. It's a great pleasure to be here this afternoon to talk to you about the next generation of AI-powered cancer visualization technology that our company, CytoVeris, is developing. All cancer patients go through immense emotional stress, and for those facing cancer resection surgery in the near future, there are 5 dreaded words that none of them want to hear. We didn't get it all. So why does this happen? Because it happens very frequently. Well, it's all about the margin. It's all about the status of the cancer margin during that procedure. And in any cancer surgery, the surgeon is faced with a delicate balance. I have to remove all of the cancer, but I have to leave as much normal tissue as I possibly can. In breast cancer, this means that the surgeon is trying to balance an oncologic outcome and a cosmetic outcome. But this sort of problem it manifests itself in just about every type of cancer surgery, and in every case, it can potentially, a positive margin, result in negative and worse outcomes for that patient. If you look at the data and the statistics on this problem, it's very clear that there's a big need to improve the precision of cancer resection surgery. If you just look at bladder cancer TURBT procedures, where small lesions are annually or very frequently removed out of a patient's bladder, those small lesions, just about 50% of those are deemed insufficient. And then those patients have then faced complications or a repeat surgical procedure. In breast cancer, depending on the institution that the patient goes to, up to 35% of the patients can be told a week later, after surgery, after they've been closed up and sent home, that we didn't get all the cancer. You have to come back for a repeat surgery. And that creates not only emotional stress, another reoperation, health burden to the healthcare system, of course, and complications for that patient in the future. And in radical prostatectomy, up to 25% of the margin cases there are positive. And this leads to additional different types of complications for those patients. Most importantly, a 2 to 3-fold increase in the chance of biochemical reoccurrence of the disease in the future. So at CytoVeris, our mission is to change that by enabling more precise intraoperative decisions, which will improve patient outcomes through the use of real-time data provided and imaging provided to the surgeon so they can see how that resection surgery is going, and from those insights provide better outcomes. So the system we've developed is called Tumor Map, and it allows for this guided cancer resection. It requires, in this system, it works on an ex vivo specimen, so we have a resected specimen out of the patient. It goes into the imaging chamber in the upper part of this system, and the system requires the patient to have no dyes, no agents administered prior to the surgery, and no dyes used on the specimen once you've had the surgery. The system basically relies on analyzing the endogenous biochemical makeup of the tissue and detecting where there's cancer present. So what we're doing there is looking at the impact that cancer has on the tissue and the changes in the biomolecular and morphological makeup local to the cancer tissue. It provides real-time information. The information is provided to the surgeon during the operational window, allowing them for it to be actionable and improve the patient outcome. And the whole system is powered on machine learning algorithms, which utilize state-of-the-art image processing algorithms, and it provides an intuitive output to the surgeon as an overlay on the tissue image. So the benefits of the system are, we believe, as we take this into intraoperative application, will be increased precision, a seamless fit of this platform with the workflow, cost savings to the healthcare system, and better outcomes overall for patients facing cancer resection surgery. The Tumor Map System, the science behind it, the technology behind it relies on a convergence of three key technology areas. An understanding, of course, of cancer biology and the biomolecular makeup of cancer, not only cancer cells, but the cancer microenvironment, the extracellular matrix, everything that's going on inside that matrix. We have developed a multispectral optical imaging technology that is very powerful. This system utilizes multispectral excitation and multispectral imaging, which essentially provides a cube of images that is very powerful in detecting the variety of different biochemicals associated with normal tissue types and also diseased tissue types. And then we feed those images into the machine learning, and that machine learning learns the patterns and the subtleties, and indeed the modulation in those biomolecular signatures to detect cancer. So this is what the system ultimately will do. This is a piece of a lumpectomy specimen on the left, shown in white light, imaged in the Tumor Map System. And the algorithm that we developed, we call Oncocite, then basically takes that multitude of different images. It's passed through the algorithm. And what we do is simply overlay a cancer heat map. Now, in this particular case, this is a section out of a lumpectomy specimen, so it's cut through the lumpectomy. So as we'd expect, the cancer, the tumor, is running down the center of that lumpectomy specimen, showing there is a good margin around it. But this is part of our training process, where we have done over 800 specimens like this and trained the algorithm to recognize cancer tissue versus normal tissue by referencing this piece of tissue and tissue like it back to the pathology H&E slides. And that allows us to get a very strong differentiation capability in the algorithm. So when we run validation tests on the algorithm by training on a subset of the data, then testing or training by leaving one patient out, we get phenomenally high performance in this system, showing that it has developed a very strong potential for recognizing cancer over normal tissue. You can see here, this is for 73 patients, over 280 targets on those different blocks. That we have a sensitivity of over 90%, a specificity that's right up at over 99%, and an overall accuracy in the system of 97%. So these are, if you like, point measurements that are made. But of course, the real challenge is converting this algorithm into a full specimen imaging to direct and guide the surgeon to where they need to take further action. So this is how it looks as when the surgeon gets to use this. On the left-hand side is, again, a white light image showing a full lumpectomy specimen. You can see the sutures, which provide some referencing for the specimen. And on the right-hand side, you can see an overlay color map, a false color map of the status of the biomolecular signatures that we detect in that specimen. The blue and the greens are cool. They show no indication of cancer. And on this, there are a couple of regions where there are red hotspots. The surgeon would be directed to those as areas that they need to look at further and explore, and indeed take further action in terms of additional excision of tissue to ensure that there's no cancer. Those additional excisions, those shavings in the case of breast cancer, can also be tested using this instrument to make sure that there's a clear margin. So with this guided tumor resection, it can lead to many, many benefits for the patient. Decreased likelihood of things such as secondary surgeries, as in the case of breast cancer. Fewer post-operative complications. A decreased need for radiation and chemotherapy. Improved functional outcome. For example, in the case of prostate cancer, of course, the surgeon is always looking to do nerve-sparing surgery. And, of course, cosmetic outcomes, top of the list, of course, in terms of breast-conserving surgery. There's a lower risk of reoccurrence and spread of the disease. And with this targeted operation, we can also look forward to more targeted therapies following the surgical procedure. The impact, of course, for patients is immense as well. If we look at that, the reduction in emotional stress and burden across the patient population, if we just reduce the number of positive margins in breast cancer surgery nationwide, it would mean that 25,000 women didn't have to go back for re-excision surgery. In prostate surgery, we'd improve the outcomes for up to 90,000 men in terms of improving the odds of preserving functionality and reducing the complications there. And likewise in bladder, 70,000 repeat procedures could be avoided by the use of this guided technology. We have developed the system and proven out the algorithm in breast. We are working in bladder and prostate as well. And for ex vivo specimen analysis, we are looking to take the technology into areas such as head and neck, where of course head and neck surgeons often use frozen section quite routinely for the small biopsies that they look at in terms of proceeding with their surgery. We believe our technology could be used as an adjunctive to that, or actually used in place of potentially that type of surgery. But the really interesting thing here is we believe this technology, while we're using a wide field imaging system in our Tumor Map platform the way it stands, the technology is implementable in a probe-based format. And we're looking to take that into various robotic and MIS applications for surgical procedures. So the markets that we're already addressing represent in excess of $1 billion market, addressable market for us over there in the European and North American markets. So a very strong and growing opportunity. And we feel that with expansion through this platform into other oncologic indications, of course, there'll be even more opportunity. But we do believe that the real value in this technology will be realized not through ex vivo specimen analysis, but with increasing trends in the use of robotic procedures for cancer surgery, building an in vivo probe-based imaging system that can provide the surgeon real-time information on the cancer margin and the residual cancer in a cavity during a robotic procedure will be really where the value is. And indeed, if you look at the robotics side of things, and we do believe that real-time molecular imaging will be one of the cornerstones of improved precision surgery. And I think that the type of technology we're developing at CytoVeris can produce transformative capabilities and accelerate adoption of the technology. Our team we have 100 years of leadership experience across the team. If you want to learn more about what we're working on and what we've done before, please contact me in the breakout room after all. I can tell you about our clinical plans and where we stand in terms of raising capital. So the future of cancer surgery as we see it is we've got it all. Thank you.